It's an honor for me to present our first keynote speaker, Dr. Thomas Givnish. Tom is a plant ecologist and evolutionary biologist and holds the Henry Allen Gleason Chair and the Willem Hofmeister Chair in Botany at the University of Wisconsin-Madison. His interests focus on the plant adaptation, speciation, molecular systematics, historical biogeography, adaptive radiation, and diversity. He and his students and colleagues analyze the connections of key physiological and morphological traits to a plant's ability to compete successfully in different contexts, and then use these connections to account for global patterns in the form, physiology, distribution, and diversity, and reconstruct patterns in adaptive radiation, geographic spread, and species diversification of plant lineages throughout time and space. That's quite a mouthful. The title of his talk is actually Progress and Prospects in Monocot Phylogenomics and Evolutionary Biology. So please, a hand for Tom.
Thanks so much. Well, got this. Okay. Thank you very much, Adam. It's a great pleasure to be here. Bienvenidos, San Jose, y MONOCOTSvii. This is the 7th International Conference on Monocot Phylogeny and Evolution. The series began in Kew in 1993 and continues today. These meetings have had an enormous impact by allowing hundreds of us to meet regularly and share our latest approaches and insights. Monocots, with 85,000 species and 77 families and 12 orders, are one of the most diverse, morphologically varied, ecologically successful, and economically important of all plant lineages. They display extraordinary variation in vegetative and floral morphology, provide most of the human diet, and comprise nearly 25% of all flowering plants. Understanding their phylogeny and patterns of morphological, geographic, and ecological evolution is thus a major challenge for plant systematists and ecologists. The Monocot conferences from Kew to Sydney, Rancho Santa Ana, Copenhagen, New York, Natal, and now San Jose have made a great contribution to meeting this challenge. Now, 2 key publications set the stage for these conferences and the attending revolution in monocot phylogenetics and the ever more powerful insights revealed by phylogenomics as we climb the information ladder, my topic today. So in 1993, the Missouri Botanical Garden, published a symposium on using sequences of the plastid gene RBCL to infer relationships among seed plants. This included an enormously influential paper by Mark Chase and 41 colleagues, and a key companion piece by Mel Duval and colleagues. Chase et al. Opened the modern era of plant molecular systematics. Its ramifications reverberate to this moment. Applying parsimony to RBCL sequences, they identified clades corresponding to many of Dahlgren's orders, but showed that others were not monophyletic and identified several new higher-level relationships. Commonilids here, and then commonilids later, appeared as a clade of 4 orders: Poales, Zingiberales, Aracales, and Commonales. One analysis in this early study by Chase et al. Placed commonilids in an odd position, sister to Dioscoriales. Duval et al., see that, obtained a similar phylogeny using maximum likelihood that placed the commonilids highlighted, as you can see in magenta, sister to Asparagales. Now, these studies both recognize something close to our current view of relationships among 10 of the 12 orders, with a few stray taxa and missing representatives of 2 orders and several families. The good news was that these 2 analytical approaches identified the same major clades and several of the same relationships among them. The bad news was that Duval et al. Found relationships among almost all orders, and many families disappeared if branches with less than 50% bootstrap support were collapsed. Nevertheless, these 2 early analyses took us important rungs up the information ladder, generating hundreds of characters from RBCL, and through community efforts and leadership by Mark Chase, scoring these across species representing several families. The nature of sequence data and its storage and access in GenBank and other repositories meant that this community effort could be expanded indefinitely by adding new taxa and new investigators. Oopsie, sorry. I'm not certain how I go backwards. Can we go back? There we go. Okay, good. A little complicated keeping my notes and the slides organized here. So at the first Monocot Conference in 1993 at Kew, what I'll call M1, Chase et al. Increased sampling from 78 to 172 species, but added no support to higher level relationships, as indicated by the arrows, relationships among the commelinids, the asparagales, liliales, dioscoriales, and pandinales all collapsed in the strict consensus tree, as did relationships among several families of elismatales. And among the commelinid orders, Poales was sister to commelinales plus zingiberales. All 3 were sister to dasypogonales, and all sister to aracales. But each of these relationships vanished in trees just 1 step longer, which indicates, I think, just how precarious the support for this phylogeny based on RBCL alone was. Now, at M2 in 1998 in Sydney, Chase et al. Added sequences for plastid ATPB and nuclear 18S ribosomal DNA, and finally reported bootstrap values for their parsimony-based analysis. The good news was that most orders had 85 to 100% support. The bad news was that most relationships among orders had less than 50% bootstrap support, as indicated by the collapsed nodes here. The low support for such relationships probably resulted because RBCL evolved slowly, and ATP, B, and ATS evolve even more slowly. And together they leave few traces of ancient phylogenetic events in the sequences of living species. Nevertheless, this paper was a real game changer, indicating a way further up the information ladder using sequences of not 1, but a few genes. It upped the ante for DNA-based phylogenetic studies, pointing to the necessity and, of course, the resulting costs of sequencing not 1, but several genes to resolve challenging relationships. Sequencing and analysis of a few genes, especially from the plastid genome, rapidly became popular, with papers in several orders, families, and genera using just this approach already in the M2 proceedings. Now at M3, at Rancho Santa Ana Botanical Garden in 2003, several more routes up the information ladder emerged. First, a team I led sequenced the plastid gene NDHF for representatives of all monocot orders in most families, producing a well-resolved tree with substantially higher bootstrap values. NDHF is 50% longer than RBCL and evolves twice as fast, so it produces 3 times as much phylogenetically useful data. And that in turn provides more resolving power and greater support for individual branches. We mapped the evolution of net venation and fleshy fruits onto the NDHF tree and found that both arose more than 20 times across monocots, often together, and almost always associated with shaded habitats, which are indicated here by the tan rectangles. The ecological correlates of trait gains and losses were highly significant and were consistent with predictions that I had made 20 years previously for net venation and Tom Crowett, sitting in the audience, had made for fleshy fruits based on functional grounds. So this case provided a vivid illustration of concerted convergence of ecologically, genetically, and developmentally unrelated traits undergoing parallel shifts in response to shared conditions to which they are adapted independently. Second, Sean Graham and his colleagues sequenced 17 plastid genes, 8 spacers, and 3 introns, totaling 16 kilobases for species representing all orders and 60 of the 93 APG2 families. Using parsimony, they produced an almost completely resolved tree with robust bootstrap support averaging 91% per branch. Positions of 4 orders, however, were not well supported. The amount of phylogenetically useful information in this study, including many long and sometimes fast evolving stretches of DNA, dwarfed that in all previous across monocot studies. It was a real step forward in approach. Third, Chase et al. Produced a synthetic analysis after the conference using plastid RBCL, ATPB, and DHF, and MACK, as well as 18S and 26S ribosomal DNA from the nuclear genome and ATP1 from the mitochondrial genome, using data contributed by the Chase, Graham, and Givnish teams. And this resulted in strong support for all orders. However, 5 relationships among orders and 28 relationships among families were weakly supported. Mycoheterotrophic taxa, which had lost several plastid loci while others evolved very rapidly under relaxed selection, were mostly excluded. Now, monocot molecular systematics, based on sequences of just one or a few genes from the plastid genome, revolutionized our understanding of higher level monocot relationships and led to their dramatic reclassification in successive versions of APG. Triumphs of this era include identifying 11 or 12 orders and 77 families, recircumscribing several groups. For example, here, finding that several genera, once placed among the lilies, actually belong to 4 different orders, recognizing several new higher order relationships, including the unity of the cotyledons, 5 disparate orders that share many nucleotide substitutions and UV fluorescent compounds in their cell walls, and the discovery that highly reduced aquatic plants in family Hydrotalaceae, once placed in Poales, based on form and RBCL sequences, are not even monocots, but basal angiosperms related to water lilies. So by 2016, the emerging consensus placed Acra sister to all other monocots, followed by the sequential divergence of the orders Elismatales, then Petrosaviales, Dioscoriales plus Pandanales, Liliales, Asparagales, and the 5 comminuted orders. However, even the most comprehensive plastid study in that now distant era, 18 years ago, based on 4 genes for 120 monocots, left many relationships unresolved or weakly supported. Such soft spots are highlighted here in the warmer colors, included relationships among the 5 comminuted orders, among several families of Poales, Zingiberales, Dioscoriales, Pandanales, and Elismatales, and among the subfamilies, tribes, and subtribes of the orchids. Mycoheterotrophic monocots, which are highlighted here with the hollow dots at the end of branches, they've gone over to the dark side, and they depend on fungi rather than photosynthesis for chemical energy. They were initially hard to place using plastid data, given the loss or rapid evolution of their chloroplast genes. Nuclear sequence data were largely excluded from studies of broad-scale relationships. Fast-evolving ITS was mostly not alignable across families, and frequent gene duplications in the nuclear genome raised deep problems regarding orthology of many other genes. But ignoring nuclear sequence data meant that we were not using the richest and fastest evolving source of genetic data to reconstruct relationships, and we were basing phylogenies only on maternal lineages, and we were blind to data that were crucial for assessing hybridization, introgression, chloroplast capture, and incomplete lineage sorting. So to address these issues, in 2008, my colleagues and I formed the Monocot Assembling the Tree of Life team, or MONOTOL, to implement a phylogenomic approach to develop a fully resolved, strongly supported monocot phylogeny based on a quantum leap in the amount and kind of data used for phylogenetic inference. Our goals were first to use next generation sequencing to obtain partial or complete plastid genomes, or plastomes, roughly 150 kilobases long, and nuclear transcriptomes, each a few megabases long, stratified across all monocot families, and then analyze them using maximum likelihood and Bayesian approaches. Second, to use the resulting phylogenies and monocot fossils to develop a new timeline for monocot evolution. And finally, to use our time-calibrated phylogenies to analyze patterns and possible drivers of character state evolution, ecological divergence, historical biogeography, and species diversification on a rigorous non-circular basis. Team members included myself, Sean Graham, Jim Liebensbach, Chris Piers, Dennis Stevenson, Wendy Zomlepper, Maria Gandolfo, Cecile Ney, Claude DePamplis, Jerry Davis, and several outstanding grad students and postdocs. Together, we produced 55 papers using this phylogenomic approach between 2010 and 2023. Now, the turn to phylogenomics was made possible by continuing Moore's law decreases in computation costs and a dramatic drop in sequencing costs with the rise of next generation sequencing. Phylogenomics allowed us and others to climb several rungs higher on the information ladder, sequencing dozens of plastid genes or even the entire plastome, and begin tapping the vast nuclear genome. Our first paper used sequences of 81 plastid genes from 68 monocots and 15 outgroups to resolve two of the most challenging questions in higher level monocot phylogeny, involving relationships among comelonid orders and among families of Poales, based on a maximum likelihood analysis of 109,000 aligned bases. We found the first strong support for resolving relationships among the comelonid orders, with Poales sister to Comelonales plus Zingiberales, and Ericales sister to Dasypogonales. Bootstrap support was 93% for the first herbaceous clade and 86% for the second woody clade. Our massive data set resolved, as you can see highlighted here, very short branches at the base of the comelonids, indicating why these branches could not be identified with certain previously using only one or a few genes. Resolving very short branches requires lots of sequence data, and the noise introduced by subsequent mutations in evolution makes the job even more challenging for branches that are both short and deep. Within the Poales, the Restiid families were sister to the Graminids. The Xyrids formed a grade at their base, and Cyperids were sister to all three groups, with Rhapadiaceae, then Typhaceae, then Vermiliaceae, sister to all other Poales. Subsequently, we used large numbers of plastome genes to resolve relationships within each order and several large families. So, for example, Craig Barrett and his colleagues used 83 plastid genes to derive a strongly supported backbone phylogeny for palms and dasypogonids. Among the palms, the climbing rattans, then the mangrove palm Nipa were sister to all other taxa. Just a year later, Barrett and colleagues analyzed relationships among families of Zingiberales using sequences for all protein coding plastid genes. Their phylogeny agreed with earlier studies in identifying the four ginger families, highlighted here in blue, as a clade, and the four banana families, shown in red, as a grade. But even including all protein coding plastid genes yielded low support for two critical branches that were both short and deep within the Zingiberales. Lam et al. used all protein coding plastid genes to assess relationships of several mycoheterotrophic monocots, placing these successfully in morphologically defined families with plastome data, despite the loss of several plastid genes and rapid evolution of others, was really a major achievement, but required sequences of a lot of plastid genes. Now, relative to this tree, which we've seen before, based on 4 plastid genes for 120 monocots from the Chase et al. 2006 benchmark data set, our 2018 analysis used RAXML analysis of 77 plastid genes for 545 monocots to produce a substantially different, substantially better supported topology. Our tree is based on 50 times as much data as the Chase et al. Benchmark. It shifts the position of 1 order and 16 of 94 families or subfamily equivalents, indicated here by the blue dots, and added 7 new families indicated by the red dots. Plastome scale data greatly increased support throughout the monocot tree. Most of the bright colors that highlighted areas of greater uncertainty at the family level in the 2006 tree disappeared in our 2018 analysis. Average bootstrap support for the branches was 97%. All orders had 100% support, as do all subfamilies and all but 2 of the 51 APG4 families represented by 2 or more samples. A few soft branches in this plastome tree remain, most notably involving the Tophili-Ace. Mycoheterotrophs, indicated by the several hollow dots, all were successfully placed and sat on branches 7 times longer than those of their green sisters. We explored the power of complete plastomes to resolve relationships in zingiberales, using 56 full circles representing all 8 families and 4 outgroups generated by Choden Sass and Chelsea Specht. We found a basal split between the long recognized clade of the 4 ginger families and a newly resolved clade of the 4 banana families. This strongly supported phylogeny, including the highly informative spacers, resolved the long debated positions of Musaceae and Heliconiaceae, at least for plastid data. Now, we used plastome data to test our hypotheses that the probability of correct branch ascertainment and level of bootstrap support should increase with the number of genes analyzed, with branch length, and with the inclusion of non-coding regions, and decrease with relative branch depth. We assessed correct, quote-unquote, branch assessment by comparing branches inferred from randomly drawn subsets of a given number of genes with those obtained by analyzing all the data. As predicted, we found that both branch ascertainment above and bootstrap support below increased logistically with log branch length and log number of genes sampled, and decreased logistically with log branch depth in highly significant fashion for all 3 predictors. Ascertainment and support were generally lowest, shown by the warmer colors in these graphs, on the shortest or deepest branches up in the upper left corner of the graphs. And lower ascertainment and support were increasingly confined to those shortest, deepest branches as gene number increased, as you can see. Similar analyses showed that inclusion of non-coding regions also increased ascertainment and support. These findings demonstrate the great power of plastome phylogenomics and show the need for such power is greatest in resolving the shortest, deepest nodes in the upper left corners of these figures. Of course, this would also apply within families, within genera as well. Now, we calibrated the plastome tree against time using 20 fossils and secondary calibration points. Monocots appear to have diverged from the common ancestor of Ceratophyllum and Eudicots 136 million years ago and began diverging from each other 4 million years later. All orders had diverged from each other by 114 million years ago. and over half the families had diverged long before the end of the Cretaceous. The most rapidly diversifying lineages were the Pacmad clade of grasses, the mostly Mediterranean climate clade of Asparagaceae, the epidendroid and orchidoid orchids, and tropical Araceae. We calibrated our plastome tree, we also used a calibrated phylogeny for Bromeliaceae based on several plastid genes to investigate patterns of evolution. This group had been challenging in the past because it has a very low rate of nucleotide evolution. So based on our analyses, bromeliads arose in the Guyana Shield, coated in dark blue, and began spreading 18 million years ago into the Andes, Central America, Northern South America, and the Caribbean. Two major invasions of the Andes, coated in red, began 15 million years ago. Hectia invaded Central America, coated in yellow, about 12 million years ago. Two invasions of the Brazilian Shield, coated in light blue, began 10 and 5.7 million years ago, respectively. We used plastid phylogenomics to produce the first strongly supported backbone phylogeny for the orchids, sequenced 75 plastid genes for 39 species, provide a backbone, and then we complemented that with 3 plastid genes that were sequenced for an additional 144 species. And the result on the right was a fully resolved and time-calibrated tree for the orchids, and you can see a major step forward on the previous consensus view of orchid phylogeny. We found that there were 3 significant accelerations of net species diversification in the orchids. The first, shown in light blue at the base of the orchidoids, the second in green at the base of the upper epidendroids, and the last in red in the pleurothalids and their allies in the Andes. Warmer colors indicate higher levels of diversification. High diversification rates are significantly associated with the rise of pollinia, epiphytism, cam photosynthesis, pollination by Lepidoptera and euglossine bees, and life in extensive tropical cordilleras like the Andes and the mountains of New Guinea. Phylogenomic analyses based on large numbers of plastid genes or entire plastomes have now become common. Many notable studies have appeared outside the Monocot program in recent years, and 3 years ago, Lee et al. Analyzed nearly 4,800 plastomes across angiosperms. The number of plastomes sequenced since has been greatly increased. Lee et al. Found the same relationships among monocot orders that we had. I have to say I enjoyed the recent paper by Vera Paz et al., who used whole plastomes to infer relationships in the large clade K of the diverse bromeliad genus Tillandsia and reconstruct its history of geographic spread in tropical America. Now, to climb further up the phylogenomic information ladder and obtain yet deeper insights into phylogeny and evolution, we need to tap into the vast sea of rapidly evolving nuclear DNA variation. There are 2 ways of doing this: using transcriptomes, the sequences of RNA being spooled off genes that are actively expressed in tissue, and DNA sequences of part or all of the nuclear genome. Using transcriptomes to infer relationships at broad taxonomic scales was pioneered by the 1,000 plant transcriptomes, or 1KP team, who developed a complex series of analyses to deal with the deep problems of homology created by frequent gene duplication in the nuclear genome, as well as, of course, transposable elements. The large 1KP team, with lead author Jim Liebensbach, published an epical paper in Nature in 2019. Their phylogeny of green plants, including representatives of just over half the monocot families, resolved the same relationships among monocot orders as our plastome tree, except that the liliales were sister to the asparagales, with both sister jointly to the commelinids. Such a cytonuclear discordance between the plastome and the transcriptome tree points to the possibility of perhaps ancient hybridization or incomplete lineage sorting deep in time in the early evolution of the angiosperms. In 2022, Timlicina et al. From the Monotol team used transcriptomes and 1kp methods to assess relationships among 192 monocot species in 72 of the 77 families using 602 conserved single copy nuclear genes. Their astral nuclear phylogeny, which is consistent with the multi-species coalescent, is again mostly congruent with the plastome phylogeny, but like 1kp, places asparagales and liliales sister to each other. Within orders, 6 of 72 families shifted position relative to the plastome tree, but 4 of those shifts involved positions that were less well supported in the plastome tree. Nuclear data strongly placed Tophiliaceae. Oops, let's go back here. There we go. Strongly placed Tophiliaceae rather than Araceae, sister to all other families of Elismatales, and presented a alternative resolution of what had been the most difficult node to resolve using plastomes. Similar results, as indicated here, were obtained using 1,375 benchmarking single copy ortholog genes or BUSCO genes, even though only 21% of those genes were single copy. A year later, Timlicina and colleagues found complete loss of transcription of 174 BUSCO genes, the nuclear genes, right? Mostly linked to chloroplast biosynthesis in 3 4 mycoheterotrophs studied. Partial to complete transcription losses, shown here, occurred in all 4 mycoheterotrophs, the genes for synthesizing hema, chlorophyll, photosystem 1, photosystem 2, and the light harvesting complex. So with mycoheterotrophy, not only do we have loss of many genes and rapid evolution in the chloroplast genome, but there's a lot of exciting things happening in the nuclear genome. And as we know, there's also interesting things happening in the mitochondrial genome. Zhang et al. Recently used transcriptomes to obtain 1,450 single and low copy genes for 610 orchid species, representing 297 genera. Their strongly supported astral tree is largely consistent with our orchid plastome tree, except with Cymbidiae, sister to Epidendriae, rather than Bandii, as we had found. Zhang et al. Used their extensive sampling to infer 14 independent origins of epiphytism, 16 origins of mycoheterotrophy, and a stunning 46 origins of lithophytism, life on the rocks. Analyzing nuclear variation by sequencing whole transcriptomes or alternatively genomes is expensive. It requires hard-to-handle RNA or lots of high-quality DNA, and involves complex downstream analyses to avoid complications due to gene and genome duplications. This approach will probably never be for everyone. But an easier alternative that still produces vast amounts of data is to sequence a subset of the nuclear genome. That's to use a reduced representation approach. For studies at or above the genus level, the most promising approach is target capture sequencing, or HiveSeq. Targeted regions are hybridized to baits made from sequences of specific regions, often relatively conserved exons, if you're looking to study a broad range of species from related species. And then the target regions are isolated and enriched by magnetic pull-down. The stray sequences are just washed away. Sequencing is then focused on these target regions, which saves a lot of time and money because individual baits are long, roughly 100 bases. HiveSeq can work with substantial divergence among the species being studied. And often can be used even with degraded DNA from herbarium specimens. Moreover, paralogs can be recaptured and then identified and analyzed properly during phylogenetic analysis. Because baits often result in successful enrichment of identifiable sites, HYPE-seq can easily be made communal with different labs producing combinable data. HYPE-seq can also often yield organellar sequences via genome skimming. Now, designing baits is the challenge. It requires genomic or transcriptomic data up front. You can design your own baits today. It requires a lot of skills. Or you can use universal baits developed by others, or you can do both. In mid-2010s, Alan and Emily Lemon and Austin Mast at Florida State produced a bait kit for 517 single-copy nuclear loci that could pull down sequences across angiosperms. And over 90 labs have working on plants have collaborated with them using this kit, though very few people working with monocots have. We have, and Nisa Karimi, and our collaborators recently used this approach to produce nuclear and plastome phylogenes for the iconic genus Callichortus from Western North America and Mexico, and use them to reconstruct character state evolution and historical biogeography, and to show that local species richness is tied not only to, as we expect, local environmental heterogeneity, but also to the diversity of mating barriers provided by different chromosome numbers in different species of Callichortus. Now, the Lemon's approach of anchored phylogenomics has made really profound contributions to our understanding of phylogeny and evolution in many animal groups. But because they kept their baits proprietary and they processed all the samples themselves, their approach for plants has not scaled. That's saying it has not led to massively parallel production and analysis of nuclear data by lots of labs across the plant systematics community. Instead, a second set of baits, the angiosperm 353 kit, designed by an international team and based on genomic data produced by 1KP, filled that role due to strategic choices by PAFTAL, or the Plant and Fungal Trees of Life project, led by Bill Baker at Kew. With PAFTAL making this kit their standard and making it available at low cost and coordinating efforts, many labs have adopted 353 and are producing an avalanche of comparable data on nuclear genes for a wide range of taxa. PAFTAL plans to produce angiosperm 353 data for at least one species in all 13,862 currently described genera. So these are very exciting times. In many ways, it's like reliving the RBCL era, but now PAFTAL has given us not one RBCL, but 353 RBCLs. Important monocot papers using angiosperm 353 have appeared for several groups, with more, including a survey across monocots in the pipeline and due to appear in the very near future. Now, I find the first papers by Perez Escobar et al. And Elliott et al. Especially exciting. Oscar and his colleagues analyzed 294 nuclear genes for 75 species and 78 plastid genes for 264 species. Their nuclear and plastome trees are very similar to each other and to our plastome tree, but with Cymbidiae, again, sister to Epidendriae, not Vandii. The pie diagrams at each node indicate the quartets supporting the main versus alternative branching patterns at those nodes. As you can see, there is substantial discordance among nuclear gene trees in the Epidendriae, along the spine of the Epidendroideae, and in the chronicidie of the orchidoid orchids. Cytonuclear conflicts between the nuclear and the plastome trees are also common in epidendroids based on this very careful study by Perez Escobar et al. Last week, Perez Escobar et al. Reported findings based on 5 times as many orchid species. These place Cymbidiae instead sister to Vandiae and both sister to Epidendriae as our original plastome tree had. Clearly, more data are needed to resolve relationships among these 3 major clades with certainty. It turns out my colleagues and I don't agree with some of this paper's conclusions regarding historical biogeography, but that's a discussion for another time and place. Analyses by Elliott et al. Of phylogenomics and parallel ecological and biogeographic shifts in poales really are stunning in scope. I felt as if I had stepped off a time machine into the 2030s when I read this paper. They generated Angiosperm 353 sequences for a species representing more than half the genera in poales and mined data, mostly plastid genes, for nearly 10,000 other species, covering more than 40% of the whole order, the largest orders, in the monocots. At the family level, their phylogeny is our 2018 plastome tree with a few tweaks. They reconstruct hundreds of dispersal events between different regions, as you can see on the lower left, and between open and closed habitats, as you can see on the lower right. And there were many parallel shifts involved in the larger families. They also mapped global variation in local phylogenetic diversity within the 6 largest families. I'm very enthusiastic about the Angiosperm 353 approach. It's going to add a lot of excitement to our meeting this week and the years ahead. I do have a few minor concerns about data management. The current release of PAFTAL, including data for nearly 11,000 accessions, contains, however, some obvious errors, like a grass sister to all other poales, or an epidendroid sister to all other orchids, and a grade of 5 orchidoids and epidendroids sister to the sister clades of the remaining orchidoids and epidendroids. Now, there are lots of potential reasons for these discrepancies, and they're absent in the papers published, but there needs to be some transparent explanation of how such issues have been resolved and will be resolved in the future, so as to maintain confidence in this approach. All right, so what lies ahead? Well, first, there we go. In the immediate future, the 10KP project, which envisions sequencing 10,000 plant genomes over the next few years, will provide huge amounts of new data to be analyzed through in silico target capture or as whole genomes. So will the Darwin Tree of Life project in Great Britain and the Genomics for Australian Plants project. I think we would all like to see similar efforts launched soon for the megadiverse floras of the US, China, and Brazil, among other areas. Now, second, the Angiosperm 353 kit needs and will be getting improvement. Its baits are based on rather few monocot genomes. And so, as you can see here with the highlights with the magenta dots, several monocot orders and families show low capture rates. Happy to report, according to Bill, that this problem will be addressed in the next release of the kit. Today's baits are also tied to slow evolving exons, which makes them great for studying broad scale relationships. The same baits work across angiosperms, right? But they're not the best for studying recently evolved or slow evolving groups. Now, that problem can easily be solved by combining Angiosperm 353 with a custom kit optimized for rapidly evolving loci in the focal group. That gets around the inevitable trade-off between fast evolving, highly informative targets and their failure to capture successfully across broad taxonomic scales. Now, making this custom kit obviously requires genomic skills, but it adds resolving power while retaining our communal ability to trace higher level relationships from the resulting data. Yardini et al. Recently did this for the slow-evolving bromeliads and developed a combined bromeliad 1776 kit. Bing Li and Jacob Lannis from my lab and from Chelsea Speck's lab have fine-tuned this approach by adding genomic data from sequences we produced representing all 8 of the bromeliad subfamilies. We added those to eliminate capture bias. We deleted less informative baits, and we added faster evolving sites to produce a bromeliad 1812 pit. I promise there will be no more wars involved in this theme afterwards. We found good initial agreement between nuclear and plastome trees in Pitcairnia, but as you can see, extensive cytonuclear conflicts in Deuteroponia. Finally, we should move quickly to phylogenomics based on whole genomes, actual phylogenomics. The current Hype-seq era that we live in may be short-lived as we transition with 3rd and 4th generation sequencing and further drops in sequencing and computation costs to producing and analyzing most parts of reference quality genomes and derive the deepest possible insights into phylogenetic relationships and plant evolution. There's a lot to be done first, however. We are currently tapping only a few 100,000 bases, the equivalent of just 1 or a few plastomes from nuclear genomes that are hundreds of millions to tens of billions of bases in size. We need to learn how to analyze large numbers of regions that are now not demonstratively orthologous, and to distinguish and deal with the effects of hybridization and incomplete lineage sorting. I feel that we may have become desensitized to how widespread these last 2 phenomena, let's say hybridization and ILS, are by our long use of plastid genes. We're all inherited as a single unit. We need to develop better genomics tools for studying both and distinguishing between them, between reticulate evolution and ILS. For example, what are the causes of the deep discordances between the plastid nuclear genes and among nuclear genes in resolving relationships close to the origins of monocots, eudicots, ceratophyllum, and basal angiosperms, revealed by Baker al. in 2022, compared with the Li et al. Studies and the Sun et al. Studies looking at chromosomal fusions. We also need new approaches to analyzing and employing phylogenetic networks, like those being developed by Claudia Solis-Lemus and Cécile Anet. The most powerful algorithms we have today cannot handle more than 20 species at a time and choke on multiple crosses between close relatives. Yes, there are some approaches that scale, but they really do not handle the complexity of reticulate evolution. Sequencing whole genomes cheaply, providing the means for resolving complex gene flow events, distinguishing them from ILS, and analyzing what we now see as non-orthologous regions are, in my view, the 4 challenges we must overcome to climb the final rungs on the information ladder to the maximally illuminating phylogenomics of whole genomes and the deeper insights that will provide us into plant evolution. Thank you all very much.
Thank you, Tom, for that wonderful talk. Questions for Tom?
Any questions?
No questions.
I've conveyed everything clearly. Oh, that could be true. Yes, Oscar.
I fear a debate coming.
No, not really. Thank you, Professor Givnish. Very impressive talk. Just to briefly clarify that the tree, I mean, we are aware of these issues. And just to clarify, by no means, I wasn't, or none of us were involved in the computation of the tree whatsoever. But the Explorer, what is being shown there is just a placeholder for what has been sequenced. That tree, it is the way it is because they use like very low threshold values to exclude data missingness, to account for data missingness.
Okay.
So things that are mostly misplaced, they are unlikely to be, let's say, related to stuff that was like wrongly sequenced or misidentified specimens, but rather specimens that are complicated and that had like very few genes retrieved, and they just got placed in like the wrong position.
Thank you. That's very helpful. I suspected something like that was going on, but as I said, it was a data management issue, and I think that it's very impressive to have 11,000 accessions up on the Tree of Life Explorer site, but it might be useful to go in and to take out the problematic sequences. And of course, they have to be taken out for good reasons, not because they disagree with previous analyses, right? In other words, there are things we're finding, right, that disagree with the plastome analyses. And that's the advantage of having the nuclear data. There are results we're finding, obviously, that differ among different nuclear genes, and that's fine. But there are some cases where it's quite obvious in terms of internal contradictions that some of the placements have to be wrong. So we have to get rid of them on a non-circular basis, not because they don't jive with our preconceived notions, but I think that can be done in terms of looking at how many genes are recovered. Turns out, for example, that there's very wide range of recovery rates with angiosperm 353. Elliott et al. Found that some of the samples for Poales had only 14% as many sites recovered, loci recovered as for other sites. And at some point, I think you have to bite the bullet and said, we're going to have to try harder on those, and we're going to drop from the analysis. We don't have that coverage because actually the insertion of samples that are missing a lot of data can distort relationships among the remaining things. Yeah. So you really have to have those taken out. Anyway, not a debate.
No, totally. Okay, just also to clarify that one cool thing of the Explorer is that all the data can be traced like to the original specimen, right? And then I know that this is based on the work of my colleagues, of course. I will be briefly talking about that during my talk. Yes. They're actually improving the bait kit because you are totally right. Even actually for families like orchids, the transcriptomes that were used as a base to produce the baits like were rather few. Yeah. So I know that they are on the works of producing a new kit.
Exactly. Thank you. Other questions? Yes.
Let's use the microphone, please, because there's people.
Thank you. Thank you for your talk.
Thank you.
Very impressive. Yes. I'm curious about how to solve deep relationships that looks like it present a lot of conflict, and we look like we solve it with some data, and then we have more data, and then we have conflicts again, and then we add more taxa, and then we solve some parts, but then we have conflicts again. You think that if we continue adding genes and adding species, that's going to resolve the problem? Or what's your view to the future? Is that the solution? We add more data, and then somehow we're going to solve it. We have to go deeper in what the data, how the data is evolving to see how to add the data, or how do you see this in the future?
Excellent question. Not just for me, but for all of you, right? Because that's the question we all have to face. And I think there are a couple things to think about. First of all, even with regard to the nuclear data, which is, we only have the equivalent of 1 or 2 or 3 plastomes of nuclear data out of hundreds of millions or billions of bases. And we know, and one of the reasons I showed the Barrett et al. 2014 study is, even if you look at all the genes, every single one in the plastome, you could not resolve with confidence the relationships among even the families of the Zingiberales, right? And so, again, the approach for angiosperm 353 is going to get some of the splash zones, some of the spacers or introns next to the exons. But really, we're still talking genes, and we're also talking genes that evolve slowly, right? That's why they're in the kit. And so I don't think we have nearly have enough data across angiosperms to be confident in the relationships presented by the nuclear tree. So that's point one. Point two is it might be that when we have a pretty consistent view, a very strong majority view from a much more expanded nuclear data set, that it conflicts with the phylogeny based on plastomes or on mitochondrial genomes. Great. And it may also be, in fact, certainly will be, that you'll see conflict among nuclear genes. So if we're expecting as a holy grail at the end, one true tree, it is never, ever, ever, ever going to happen, right? And the reason is that evolution, while it may be mainly or mostly tree-like, is also reticulate. You have networks, and I think we have to wrap our mind around that possibility. We also have to wrap our mind around the reality of incomplete lineage sorting. When you have rapid diversification in a lineage, often you do not get the proper, quote unquote, sorting of variants in different genes tracking the cladogenetic events. I think everyone in this room probably has written a paper that said, well, this conflict may be due to conflict between due to conflicts introduced by hybridization or ILS. Almost all of us have done that. We have to stop repeating that piety and start actually running analyses which are available today to see if we can exclude, say, ILS in several cases. So, for example, in the case I mentioned for Pichiernia, we're able to exclude all but one of the reticulation events, all but one of the seeming reticulation events as being due to ILS. So ILS is not important. There is some actual hybridization or introgression in the past. And as I said in my talk, the problem today is that our data are now ahead of our algorithms, right? We don't have enough power with the algorithms per se, but also with the computers to take up fairly complex but entirely reasonable scenarios. And I know Claudia, I know Cecile, they're both at UW. They're both very, very bright people, and they're working hard on it, but they're not there yet. And I don't think the field is there yet. So we're going to have to wait for that. Is that answered? Okay, very good. Yes, Sean.
Sorry, now there's 2 or 3 questions. Guys, we have time for, I think, 1, maybe 2. I'll go here and maybe...
I'll try to make my answers shorter.
So thank you, Tom. So we think a lot about conflict because those are the things that bug us, but I'd like to point out congruence and how much there is across everything that you talked about today. It's phenomenal, and it's really reassuring. I like the idea of coming back from monocots 8 or 9, when we have genomes for everything and seeing, what happens then. And I agree with you that thinking about ILS and hybridization, coming up with better methods for those would be is really important. I think we're probably going to turn into thinking more about using phylogenies to think about genomes rather than the other way around. My question, though, is that I wanted to sort of point back to when did Chase it all come out? So that was one gene.
1993.
1993, that sort of era.
Right.
And it's just, that gene and maybe ATPB are sort of, that's the basis of much of our classification of angiosperms. And just, it's pretty remarkable to me how much they got right. And I wondered your thoughts, sort of looking the other way around, about why single genes worked so well.
Yeah. That's another excellent point. So, in fact, our views have changed. Our 2018 paper shifted around 16 different entities and the relationships of 2 orders. So the changes were not trivial. I think it's remarkable, though, considering that the phylogenies at the family and ordinal scale had almost no support, right, just based on RB-cell. And really, adding ATPB and whatever, you just 18S. You have to say, how lucky were we? But I think what happened is the pattern was there, and that was the most likely pattern to pop up even in parsimony analysis. And one thing I'll mention, since you asked the question, Sean, is that I didn't say anything about mitochondrial genomes. I had a section on there, but I felt I was going to run too long. And I just want to point out how, while mitochondrial genomes generally have been avoided because they're slow evolving in plants, you and your colleagues did a great job placing a lot of the mycoheterotrophs in morphologically defined families. And you're going to talk about it this afternoon, right? Oh, this morning. Coming up soon. Last question.
Hi, thank you very much, Tom, for an amazing talk. I wanted to ask you, if we move towards this whole genome phylogenomics and the issues of hybridization will become more and more apparent, you pointed out that we miss some of the tools necessary, but I was also wondering what happens, because we did this with Tillandsia, for example, with whole genomes, and then we start to see sections or clades which are 5% introgressed, 10%, 20% introgressed with the other section, other clades. So how do you see this? How will this transform, our understanding of phylogenetics in monocots?
Again, excellent question. I think in two ways. One is just a lot more data, analyzed the same way we're analyzing today. Actually, three ways. No one expects the Spanish Inquisition. So a lot more data. Secondly, I think there are some key cues to hybridization, like inheritance of syntenic blocks from from individual parents, that'll become much more important at a different level of analysis than we can possibly resolve now using the data as they're analyzed now. And then finally, even within chromosomes, you'd expect linkage, right, to decline exponentially with distance among sites. And I think it's important to basically be able to analyze conflict as a function of linkage, right, as a function of distance on the chromosomes, or whether the things are on different chromosomes. So I think that, I didn't put that in my talk, but those were the things I'm thinking about in terms of how we could move forward.
All right, thank you, Tom, so
My
much. Yeah.